Welcome to This Explainer. Today, we're jumping right into a genuinely groundbreaking 2025 study from the journal Frontiers in Pharmacology. We're looking at a powerful, entirely new approach to combating chemotherapy-resistant ovarian cancer. We're going to get right down to the microscopic level to see exactly how targeted botanical therapies can intelligently dismantle aggressive tumors. Here's our agenda for today. We'll start with the clinical challenge, then uncover the one-to-one synergy breakthrough, explore its selective safety window, see how it deactivates the tumor engine, look at halting metastasis, and finally, look toward a new horizon in oncology.
Section 1. The Clinical Challenge.
Why is ovarian cancer so difficult to treat?
You know, ovarian cancer is notoriously silent, and that all too often leads to late-stage diagnosis. The really frustrating reality for oncologists is that standard platinum-based chemotherapies often hit a massive rigid wall. These tumors adapt. They develop a fierce resistance that drives recurrence. So how can modern molecular oncology actually overcome this resistance in such aggressive tumors? Well, researchers are now shifting their focus toward combining active botanical compounds, specifically cannabidiol or CBD and tetrahydrocannabinol or THC, to completely bypass those ingrained defenses.
Section 2. The one-to-one synergy breakthrough. The power of precision.
So it turns out the crucial detail here is an exact equimolar dose, 2.5 micromolar of CBD to 2.5 micromolar of THC. In this study, researchers tested all sorts of concentrations and ratios on both platinum-sensitive cells and innate platinum-resistant cells. And the results were honestly staggering. This exact one-to-one ratio creates a highly synergistic attack that far outperforms using high doses of just a single compound. It's not about just blasting the cancer with higher chemical concentrations. It's about the incredible precision of these two compounds working in tandem to crack the tumor's armor.
The math behind this synergy is absolutely fascinating. When calculating the Chow-Talale combination index, a value of 1.0 means two drugs simply add together. Anything above 1.0 means they actually interfere with each other. But with this precise one-to-one ratio, the index drops well below 1.0. hitting values as low as 0.3 and 0.5 at higher effect levels. That provides undeniable mathematical proof of a powerful synergistic cytotoxic effect against these ovarian cancer cells. In this case, the sum is literally far greater than its parts.
Section 3. Selective safety window. Protecting the healthy.
Now, killing cancer cells is really only half the battle, right? We absolutely have to avoid collateral damage. Picture this incredible contrast. The aggressive cancer cells are essentially dissolving under this treatment, with their viability dropping sharply at tiny doses, an IC50 of just 4 to 6 micromolar. Meanwhile, the healthy ovarian epithelial cells remain virtually untouched, acting like they have a cellular shield around them. They require more than 21 micromolar, which is over 4 times the concentration, to show any significant negative impact. Finding a selective safety window like this is basically the holy grail of oncology.
Section 4. Deactivating the tumor engine, the molecular mechanism.
To truly understand how that selectivity is even possible, we have to look intracellularly. Think of the PI3K, AKT, and mTOR signaling axis as a massive, hyperactive engine. In ovarian cancer, this specific pathway is aggressively fueling tumor growth, it's blocking natural cell death, and it's keeping those cancer cells alive even when they take direct hits from chemotherapy. It is literally the survival engine of the tumor.
Well, the one-to-one CBD to THC combination executes a pretty brilliant two-step maneuver to destroy that engine. First, it completely cuts the fuel. It does this by shutting down the phosphorylation, or the chemical activation, of those PI3K, AKT, and mTOR proteins. Second, and this is maybe the most amazing part, it simultaneously restores the cell's natural brakes. It upregulates a tumor suppressor protein called PTN. This acts exactly like pulling a glowing red emergency brake, stopping the cancer's hyperactive growth cycle right in its tracks.
And the Western blot data gives us the hard physical evidence of this mechanism actually happening. When analyzing the protein bands, the control row confirms we have equal amounts of protein across the board. But when evaluating the phosphorylated proteins, the activated ones, under the combination treatment, those dark bands vividly fade away. The survival engine is shutting down. At the exact same time, the band for the total PTN protein lights up brightly, proving that the critical braking mechanism has been successfully restored. With the engine totally deactivated, it is absolute chaos for the cancer cell. Flow cytometry data reveals exactly what happens to the cancer cell's mitochondria.
In healthy cells, specific dyes form red polymers, but under this synergistic combination, there's a massive shift to green monomers. What that means is severe mitochondrial depolarization. The mitochondria are losing their structural integrity, which causes a huge spike in reactive oxygen species, creating overwhelming oxidative stress. And that stress, it triggers apoptosis, which is the cell's natural programmed self-destruct sequence.
Section 5. Halting cancer metastases, blocking the spread. Beyond just forcing cell death, we have to look at the cancer's lethal ability to actually spread through the body. The data shows a complete crippling of the cancer's invasive capacity. In clonogenic assays, we see the combination therapy utterly destroys the cancer's ability to form long-term reproductive colonies. The cell clusters just vanish. And in transwall migration assays, where cells attempt to invade through a matrix, the migration is almost entirely blocked by the treatment. It's as if the cancer cells hit an impenetrable energy barrier. Section 6. A New Horizon. Summary and Impact.
So stepping back from all the dense molecular biology for a second, the executive takeaways here are pretty definitive. This precise one-to-one equimolar combination isn't just randomly attacking cells, it is a highly targeted intervention. It significantly decreases migration and invasion. It aggressively increases programmed cell death and oxidative stress. It intelligently shuts down those oncogenic survival engines while fixing the natural brakes. And incredibly, it does all of this while safely bypassing healthy, non-tumor tissue.
As researchers push forward with in vivo validation to see exactly how this translates from the lab bench to a living body, you really have to wonder, are we looking at a genuine paradigm shift? Could precise botanical synergies like this be the key to unlocking the next generation of integrative targeted oncology and fundamentally rewrite how we treat the most resilient cancers? It is an absolutely fascinating frontier. Thank you so much for joining me on this deep dive. Keep questioning, keep learning, and I'll catch you in the next explainer.